Dear organization team, dear participants, first of all, I'm very happy to be here with all of you. Let me introduce myself. My name is Narimane Segueni. I'm professor at the Faculty of Medicine, University Saleh Boubnider, Constantine III, Constantine, Algeria. My talk today is entitled Anti-Cancer Properties of Algerian Propolis, Experimental Evidence, Mechanism, and Future Perspectives. Bee products are valuable and rich source of biologically active substances. They have been used for centuries in traditional medicine in the treatment and prevention of many diseases. Propolis has been gaining attention. Propolis is a natural resinous complex material produced by honeybees from substances collected from birds, leaves, and exudates of different plants and trees. The collected substances are mixed with pollen, wax, and enzymes secreted from bees. The chemical composition of propolis is quite variable. It depends on several factors such as geographical and botanical origin, the method used during collection, the time of collection, bee spaces, and many other factors. Algerian propolis has recently begun to be studied. Therefore, information concerning its chemical composition, botanical origin, and therapeutic properties are still very limited. Regarding the chemical composition, up to date, the different research have led to the identification of two main types of propolis in Algeria. The first type is a popular type, and this type is rich in phenolic compounds such as caffeic esters and flavonoid. And the particularity of Algerian propolis is that our propolis is very rich on caffeic acid and its derivative, caphtoric acid and its derivative, chichoric acid and its derivative, and we isolated a lot of them in our laboratory, and our investigation demonstrated that those compounds possess very interesting pharmacological properties. Here you can see some of the isolated compounds from popular type propolis in Algeria, and the research demonstrated the isolated of these compounds for the first time from our propolis, suggesting the use of this plant as a source of our propolis. The second type identified in Algeria is a Mediterranean type propolis, and this type is very rich in D-terpene compounds, in particular those belonging to labdan and clerodan family. And these propolis, or those propolis, were also found to be rich on polymethoxyflavonol. And here you can see some of the isolated compounds from this propolis, suggesting the use of Citrus sp as botanical origin. In addition to flavonoids and phenolic acids, our propolis were also found to be rich in terpenes belonging to different skeleton, such as triterpene, monoterpene, sesquiterpene, and we also found or isolate or identify other compounds in our propolis, and those compounds were fatty acids, esters, sugars, and many other classes of compounds. And here you can see some of the flavonoids that has been isolated and identified in our laboratory. As you can see in these slides, our propolis were very rich in flavonoids, and some of K components in our propolis were chrysin, galangine, tectochrysine, pinocombrine, pinobonxine, conferol, acacetine, pinostrobine, and many other compounds. Regarding phenolic acids, our research indicated that our propolis, in particular those collected from the northeast of Algeria, were very rich on phenolic acids. And the two main phenolic acids we identified and isolated in our propolis were ferulic acid and caffeic acid. And here you can see some of the terpene isolated from Algerian propolis. Those terpene has been isolated from Mediterranean type propolis. We also investigated in our laboratory pharmacological properties of Algerian propolis, and you can see some of them. We studied the antioxidant, antibacterial, antibiofilm activities in addition to wound healing activity, anticholinesterase activity, and the potential modulation of matrix metalloproteinase by Algerian propolis. And today we will focus on the anti-cancer activity of our propolis. So cancer is a major cause of death. Lung, colon, breast, and prostate cancers are the most prevalent type of cancer. Current therapies involve surgery, radiotherapy, and chemotherapy. The growing tumor resistance to the available treatment has become a problematic issue. Therefore, there is a lot of interest in efforts to discover novel anti-cancer drugs with less toxic effects and more sensitive to cancer cells. Propolis is a natural hive product possessing a large spectra of therapeutic activity, and its use as a complementary and alternative medicine in several diseases is growing all over the world. Many research have reported its effectiveness in the treatment of cancer by acting on different signaling pathways involved in the regulation of different cellular processes. However, little is known about anti-cancer activity of Algerian propolis. Algerian propolis anti-cancer properties have recently been investigated in a variety of cell lines, including breast, pancreatic, lung adenocarcinoma, colorectal cancer cells lines, and melanoma. We reported in the present presentation experimental evidence of the reported anti-cancer properties, mechanisms of action, and future perspective. Algeria is situated or located on the north of Africa, and it is one of the biggest country in Africa, and we got three different climates in Algeria. In the north, in the coastal area, we have a Mediterranean type climate. In the south, we have a Saharan climate, and between the two, we have a sub-Saharan climate. And we have a great diversity in flora. That means that Algeria is probably or likely to produce or have or possess interesting properties in these regards, with an interesting chemical composition and, of course, an interesting pharmacological properties. So, due to the lack of time, we'll just see results concerning two types of cell line cancer, breast cancer and pancreatic cancer. For breast cancer, we choose a bio-guided approach in this research, and this approach leads to the choice of the most active propolis, which we perform on this propolis more investigation in both anti-cancer properties and also chemical composition. And this propolis was collected from the wilaya of Mila, located on the northeast of Algeria. First of all, we extract the biological active compounds using solvent of variant polarity, then we investigate the total phenolic and total flavonoid contents. We also perform FTIR analysis. For anti-cancer activity, cell viability was assessed by the MTTC assay, the MTT assay on human breast adenocarcinoma cells. We also perform apoptosis analysis. And to determine the effect of our propolis on the level and gene expression of some K enzyme, such as caspase-3, BAX, BCL2, CDK2, and P53 protein, in addition to a lot of other propolates, Western blot analysis and real-time PCR were carried out. Our results indicate that total phenolic and flavonoid contents were varying significantly according to the used solvent during extraction. And as you can see in this table, methanolic extract was found to be the richest extract in those compounds, so phenolic and flavonoid compounds, while petroleum ether extract was the poorest extract on those compounds. FTIR analysis of 3 of the studied extracts, chloroform extract, ethyl acetate extract, and methanol extract, seem practically superimposable, which means that the 3 tested extracts present the same chemical profile. And this propolis was rich in flavonoid and other aromatic compounds, aliphatic compounds, alcohol esters, and sugar. Those results were confirmed with our investigation of this propolis. We performed a phytochemical investigation, and this phytochemical investigation of all those extracts lead to the isolation and identification of several flavonoid and phenolic acids, but results are not reported here in the present presentation. So on the contrary, the 4th extract, which is petroleum ether extract, was completely different, and the FTIR spectrum of this extract was different from the 3 other extracts. This extract was found to be rich in fatty acid compounds. Regarding the MTT assay, our results indicate that the extracts were found to efficiently inhibit the metabolic activity of the tested cell line in a dose-dependent manner. And here you can see the EC15 value obtained, and those values were ranging between 97 and 117 microgram per milliliter. Regarding the expression of the K protein and gene, our results indicate an increase, and this increase was significantly observed between the tested extract on the expression of caspase-3 and the P53 protein. We also observe a decrease in the expression of BCL2 and CDK4. For the expression of BAX, the expression was different according to the tested extract. Here you can see the results of the gene expression on the treated and control cell. And as you can see, we observe an increase in the expression of caspase-3 and P53 protein, a decrease in the expression of BCL2 and CDK4, and for BAX expression depends on the tested extract. Regarding apoptosis analysis, After 24-hour treatments, apoptotic cell proportion were increased significantly. The ratio of apoptotic cell population in treated cell was nearly 6 to 10-fold higher than the control cell population. Results of the present study showed that Algerian propolis possesses an interesting cytotoxic activity in the breast cancer cell line. The tested propolis probably induced an intrinsic pathway of apoptosis through caspase cascade and activation of pro-apoptotic protein in the tested cell line. In addition, cell proliferation was found to be inhibited by the diminution of cyclin-2 and CDK4 activity associated with an increase in P21 actin as a protein inhibitor. The methanol extract was the most active extract. The tested propolis was a popular type propolis. Therefore, the observed cytotoxic effects can be attributed to flavonoids and phenolic acids, which are the main constituents of such propolis type. Other research have been performed on pancreatic cell line, and the results of those research indicate that the studied propolis cause a significant potentiation of doxorubicin cytotoxicity. Algerian propolis was combined with doxorubicin, and this combination dramatically increased the sensitivity of the cell tested to doxorubicin. The used dose was 100 microgram per milliliter, and this dose was very interesting, and it caused a decrease in EC15 by 10-fold. This potentiation was more effective than the used control, and the used control in this study was verapamil. Another result, the studied propolis caused an induction of cell cycle arrest, particularly in G0, G1 and sub-G1 accumulation. And these results indicate that it was, or we observed, or the researcher observed a substantial increase in apoptotic or senescent cells. Another result is that the studied propolis enhanced apoptosis induction. This propolis, when it was used alone, induces apoptosis with a high, very, very high level, and its combination with the doxorubicin maintained a very high level of apoptosis, and this was confirmed by the studied, and this apoptosis was dependent on caspase activation, in particular caspase 3 and caspase 9 activation. The tested propolis inhibits also P-glycoprotein activity, leading to increased intracellular drug accumulation. So pre-treatment with propolis increased intracellular DOX accumulation in the treated cell, and propolis was more effective than the used control. And as I mentioned before, the used control in this study was verapamil. Finally, the tested propolis increased rhodamine retention, and these results strongly indicate that Algerian propolis effectively inhibits P-glycoprotein pump activity, preventing the cancer cell from expelling doxorubicin and thus increasing its intracellular concentration and efficacy. Algerian propolis acts as a powerful chemosensitizer for doxorubicin in pancreatic cell line. The mechanism of action observed were directly contribution to the cell cycle arrest and apoptosis, inhibition of the P-glycoprotein drug efflux pump, thereby overcoming a common mechanism of multi-drug resistance and allowing higher intracellular concentration of doxorubicin to exert its cytotoxic effects. And the third mechanism of action was that propolis was more effective than verapamil in rondamine assay. For the other type of cell cancer, Algerian propolis has demonstrated anti-cancer effects against a range of cancer cell lines, including breast cancer, as I mentioned before, pancreatic cancer, glioblastoma, colon cancer, prostate cancer, and leukemia, and many other type of cell line. And mechanism identified up to date related to the study performed on Algerian propolis were summarized here. And as you can see, the main mechanism were induction of apoptosis involving caspase activation, inhibition of cell proliferation and cytotoxic effects, and causing a cell cycle arrest. And this was generally observed in the G0, G1 phase, preventing cancer cell from dividing. As a conclusion, Algerian propolis is a rich source of bioactive compounds with significant potential for cancer prevention and therapy. The anti-cancer properties of Algerian propolis are involved in multiple pathways. Most identified and studied are apoptosis and cell cycle arrest. Further research to isolate and characterize active constituents using a bio-guided approach and evaluate their efficacy and safety in vivo are in need. In those regards, or in those study, or in this approach, we have to focus on the synergetic combination to determine whether the observed effect was due to a single compound or a synergetic action of multiple compounds, as it is often the case with natural products. As a future perspectives, lots of work have to be done to help to better understand the anti-cancer activity of our propolis and also to potentially use it in near future. So we have to deeper study the mechanism of action to identify specific molecular targets modulated by Algerian propolis, its isolated compounds or synergetic compounds or group of compounds responsible for this activity. We have also to investigate the effects on angiogenesis on human cell and modulation also. We have also to perform study on pharmacokinetic and pharmacodynamic to determine the absorption, distribution, metabolism, and excretion profile of our propolis, in particular active compounds, as well as their bioavailability and optimal dosing regimens. We have also, we must also conduct comprehensive acute and chronic toxicity studies to establish safety profiles, to validate the synergetic observed in vitro between doxorubicin and propolis. This synergetic effect was observed in many cell lines, not only on the pancreatic cell I reported here, but on many other cancer cell lines. And these results have to be validated by in vivo study on different animal models, assessing both enhanced efficacy and potential reduction in chemotherapy-associated side effects. And we also aim, hope, and want also to work on the development of a standardized extract of our propolis, and why not find a new formulation of these extract using new technology to get the best activity with the best formulation. Before to leave you, I want just to show you some beautiful photo of my city, Constantine. So Constantine is located on the northeast of Algeria. My city is called the City of Bridge. It's a very old city with a very interesting history back in the 200 of years ago. And we got 7 bridges in Algeria, in Constantine, I'm sorry. And those bridges are linking the different parts of the city, because my city is one of the most highest city in Algeria. It is built on rock, and the different part of the city are linked by the bridge, and some of them were built in very old time, and some are new, as you can see here in the photos. For any contact, you can send me emails to one of the emails you can see here. And finally, thank you for your attention.